Assalamualaikum and salam sejahtera. We are from Group 1. We will be presenting about the microbial pathogen in food, which is Salmonella typhii.
Me, myself, Nur Fadila binti Muhammad Alwi with metric number A200353. Today, I will discuss about the introduction to the pathogen, which is Salmonella typhii. Salmonella typhii is the bacterium responsible for the typhoid fever. According to the World Health Organization (WHO), typhoid fever is a severe and a potentially life-threatening illness. It's primarily transmitted through the consumption of contaminated food or water. Once ingested, the bacteria multiply and spread into the bloodstream. Salmonella typhii is a gram-negative, obligate anaerobic bacterium that exclusively infects humans. It has no natural reservoir outside of humans, and its transmission occurs through the fecal-oral route. This pathogen causes systemic infection targeting the lymphatic tissues, liver, spleen, and bloodstream. The disease is prevalent in the areas with poor sanitation, especially in developing countries, affecting an estimated 21.5 million people annually. Historically, typhoid fever has caused significant outbreaks, including one in Jamestown, Virginia, during the 17th century. Next, I will explain about the characteristics of the Salmonella typhii. First, it belongs to the serogroup D within subspecies 1 of the Salmonella genus. 2. Strain differentiation method, which is differentiation can be done using techniques such as phage typing, pulse field gel electrophoresis (PFGE), and ribotyping. Third, antigenic properties. Possesses specific antigen, which is O antigen, H antigen, and VI antigen, which significantly contributes to the virulence.
Next, Salmonella typhii is known for its antibiotic resistance, including MDR, which complicate treatment efforts. Its genome size range from 3.9 to 4.9 megabase, with numerous pseudogenes pointing to recent evolution. The bacterium also exhibits significant genetic variability through insertion, deletion, and horizontal gene exchange, contributing to its adaptability and persistence. My name is Nur Farhana binti Azizan. My metric number A200314, and I will explain about the Salmonella typhii related to the food safety. So, Salmonella typhii causes typhoid fever with symptoms like high fever, abdominal pain, and intestinal complication. It spreads through the fecal-oral route, contaminating food and water via fecal method. Unlike other Salmonella species, it exclusively infects humans and can be life-threatening if untreated. Preventative measures including maintaining clean water, proper waste disposal, regular hand washing, and safe food handling. Practicing this measure is crucial to reducing typhoid fever risk and ensure the food safety.
Assalamualaikum and good afternoon. My name is Nurul Atika Auni and my metric number is A187548. Now, I will be presenting about the toxins and pathogenic metabolites produced by Salmonella typhii. Let's start with endotoxins. These are lipopolysaccharides, also known as LPS, that are found in the outer membrane of the bacterial cell wall. When the bacteria die, the cell walls break down and these endotoxins are released. Once released, they trigger strong immune responses in the host. This immune response often results in inflammation and fever, which are common symptoms of Salmonella infections. Next, we have enterotoxins. These are proteins that Salmonella produces during its growth phase. Enterotoxins are responsible for causing fluid secretion into the intestines, which leads to diarrhea. Additionally, they disrupt the electrolyte balance in the body, contributing to further complications in infected individuals. Finally, we have cytotoxins. These toxins directly damage host cell by interrupting protein synthesis. When this happens, the affected cells die, leading to tissue damage. This is what we refer to as cytotoxicity, and it significantly contributes to the severity of Salmonella infections. Now, let's discuss these toxins' response to heat. Firstly, we have heat-labile toxins. These toxins are sensitive to heat and can be destroyed when exposed to temperatures of 100 degrees Celsius. For instance, the enterotoxins produced by Salmonella fall into this category. This means that cooking or heating food to high temperatures can eliminate these toxins and reduce the risk of illness. In contrast, heat-stable toxins are resistant to heat. Even after cooking, these toxins remain toxic. A common example of heat-stable toxins is those produced by Staphylococcus aureus. This explains why, despite proper cooking, food can still cause illness if it contains heat-stable toxins.
Hi, my name is Wang Xingyi, matrix number A2000232. I will further explain about the toxin and metabolites produced by Salmonella. So I'm pretty sure everyone has heard about the mandatory typhoid vaccine required when applying for jobs in food handling. But what exactly causes typhoid fever? Typhoid fever is caused by Salmonella enterocerora typhii, a bacteria that produces an AB toxin. This toxin consists of an enzymatic A subunit and a receptor binding B subunit. Once inside the host cell, specifically within the Salmonella containing vacuole, the bacteria secretes the toxin into the acidic lumen. The toxin is then transported to the extracellular space via vesicle carrier. This process involves cellular components such as sorting receptor and a protein that facilitates fusion with the plasma membrane. Typhoid fever can present with moderate to severe symptoms, and if left untreated, it can lead to complication to even death. The pathogenicity of Salmonella typhii is closely linked to its ability to manipulate host metabolic pathway, particularly in macrophage, to enhance its survival and replication. One key mechanism involves inducing a metabolic shift in host macrophage, such as increasing glucose uptake and enhancing glycolysis. This creates an environment rich in glucose, which the bacteria uses as nutrient source for intracellular replication. Studies have shown that patients with typhoid fever exhibit increased glucose level and higher risk of glycolysis, of it which significantly contribute to bacterial virulence.
Hi, my name is Zainab Azali, 8100179. Okay, today I will talk about the factor affecting toxic production interesting for Salmonella typhii. The first factor is pH. Salmonella typhii needs 6.027.5 pH to produce toxin. This is because it is the optimal pH where enzyme activity, more efficient synthesis at the slightly acidic or neutral pH. At this pH, production toxin will be high. Also, Salmonella typhii cannot produce toxin if the pH is below than 4.6. If because the pH is too acidic, it will reduce and inhibit the rate of growth of the typhii and the toxic potential will be limited. Next, the factor is water activity, where according to APIA M2020, Salmonella typhii needs 0.93 to 0.95 water activity to produce toxin. This is because the Salmonella typhii insect needs 0.91 water activity to grow. If the water activity is more than 0.93, the metabolic process can be fully functioning, whereas the rate of the growth Salmonella typhii will be increased and this makes the toxin production also increase. The factor is nutrition ability, where in food or water that contain all the nutrition that Salmonella typhii needs. Example is a protein that contain protein, lipid, carbohydrates, vitamin, and minerals. All of these nutrition ahead of growth Salmonella typhii also help in enzymatic production, provide energy, present membrane synthesis, and also increase the Salmonella activity, whereas it will help in increase the toxin production.
Now, I will explain the extrinsic factors that influence the microbial toxin in foods, which is focusing on Salmonella typhii. The first factor is temperature. Salmonella typhii is a mesophile, growing optimally at temperatures between 35 to 37 degrees Celsius, which is similar to human body temperature. So, improper food storage at room temperature allows the bacteria to multiply rapidly. Keeping temperatures below 4 degrees Celsius slows its growth, while cooking above 70 degrees Celsius kills Salmonella and inactivates any toxin. Second factor is humidity. High humidity levels in storage areas promote moisture availability, essential for Salmonella growth. Food stored in moist conditions create a suitable environment for the bacteria to grow. The third factor is atmospheric oxygen. So Salmonella typhii is a facultative anaerobe, which means it can grow both in the presence and also absence of oxygen. Next is poor storage and handling practices, which can significantly contribute to Salmonella contamination. For example, leaving food out at room temperature for extended periods, cross contamination during the food handling using unclean surfaces or tools, and also improper packaging such as damaged containers, exposing food to moisture and also oxygen.
Hello everyone, my name is Nur Shahada binti Shahar Effendi and my matrix number are A1922719, and today I will walk you through the mechanism of infection caused by Salmonella typhii. Let's take a closer look at each stage. The infections start with the contaminated food or water, often due to the bacteria shed by infected individuals through feces or urine. Poor sanitation, sewage contaminated water, and inadequate hygiene are key contributors. Contaminated items including raw produce are irrigated with untreated wastewater, undercooked shellfish, and improperly handled food serve as pathway for the bacteria to infect humans. Next is the second stage, which is transmissions to the host. Transmission occurs when a person ingests contaminated food or water. Once in the body, Salmonella typhii survive the acidic stomach environment because of its acid tolerance mechanism. The third is the invention of the intestinal lining. Here, the bacteria start by adhering to the epithelial cell in the small intestine. From there, they invade arm cells, specialized immune cells in the intestinal lining to cross the intestinal barriers. Inside, macrophage engulf the bacteria. However, instead of being destroyed, the bacteria exploit these immune cells to spread throughout the body, causing systematic infection and the symptom of typhoid fever. Thank you.
After entering the host, Salmonella typhii infects intestinal epithelial cells and is engulfed by the microphage as part of the immune response. However, instead of being destroyed, the bacteria use a specialized mechanism called the type 3 secretion system, T3SS. This system injects bacterial proteins into the host cell, disrupting normal immune processes. Specifically, T3SS blocks the fusion of the phagosome, where bacteria are trapped, with lysosome, which contain destructive enzymes, allowing the bacteria to avoid destruction. Inside the macrophage, Salmonella typhii survives and multiplies within a modified compartment called the Salmonella containing vacuole (SCV), using host nutrients for growth and replication. Once Salmonella typhii has multiplied inside macrophage, it uses these immune cells as transport vehicles to spread throughout the body. The bacteria are carried to key organs like the liver, spleen, bone marrow, and gallbladder, where they colonize and replicate further. After reaching these sites, the bacteria re-enter the bloodstream, causing bacteremia. This systemic spread leads to characteristic symptoms of typhoid fever, including prolonged high fever, abdominal pain, and systemic inflammation.
Assalamualaikum, my name is Nur Hafiza binti Azmi. So for the last part of the mechanism of infection by Salmonella typhii, I'll be touching on disease manifestation. So after the bacteria grow and reabsorb back to the bloodstream, this is where the symptom will start to appear in infected person. So after the incubation period, around 7 to 14 days, the symptom will appear such as high fever, abdominal pain, diarrhea or constipation, dry cough, raw spot or rashes. But there's a severe cases where the bacteria persists in the gallbladder. Next, for the transmission continuation of this disease could happen through bacteria shedding or Salmonella typhii shedding, where the individual who harbor this bacteria could shed this bacteria through the feces, which contributing to the spread of the disease. By that, close contact with the infected person, especially in a poor hygiene setting, could increase the risk of disease transmission.
Hello everyone, my name is Nina Adriana binti Shamsuddin and my matrix number A195829, and today I'm going to talk briefly about a type of disease caused by Salmonella typhii, which is a typhoid fever. So this infection primarily spread through contaminated food and water, especially in areas with poor sanitation. So what is typhoid fever? Typhoid fever is a systematic infection characterized by prolonged fever, bacteremia, and inflammation of GI tract. Okay, next I'm going to talk about symptoms of typhoid fever. So on the first week when someone gets infected, it is called initial symptoms. The onset of typhoid fever begins with mild symptoms like fever, weakness, and fatigue. Other early signs may include headaches, loss of appetite, abdominal discomfort, and sometimes a dry cough. Next is the progressive symptoms on the week 2 to week 3. If untreated, the symptoms getting worse. Patients may develop a persistently high fever, severe abdominal pain, diarrhea or constipation, and a distinctive rose-colored rash on the abdomen. Mental confusion and agitation may also occur as the disease progresses. And lastly, severe cases. In severe cases, typhoid fever can lead to life-threatening complications such as intestinal perforation, severe bleeding, and infection spreading to other organs like the heart or brain. These complications require immediate medical attention. Fortunately, typhoid fever is preventable and treatable. Good hygiene practices, clean drinking water, and vaccination are key to prevention. Early diagnosis and antibiotics are critical to managing the disease effectively. That's all for me. Thank you.
Good day everyone, my name is Fazana Shaira binti Faisal Isham, A195956. We'll be presenting on the population risk of Salmonella typhii. Salmonella typhii affects specific population more severely due to environmental, biological, and lifestyle factors. First, let's look at geographical risk. The disease is most prevalent in low-income regions with inadequate sanitation infrastructures, such as South Asia and Sub-Saharan Africa. These areas face the highest burden of typhoid fever due to poor access to clean water and proper sewage systems. Next is age groups. Children and adolescents are particularly vulnerable because of their immune system may not be fully developed. Additionally, adults with compromised immunity, such as the elderly or those undergoing medical treatments, are also at significant risk. For immunocompromised individuals, the danger is even greater. Those with weakened immune systems due to conditions like HIV, AIDS, malnutrition are more likely to experience severe symptoms and complications. Finally, travelers to endemic areas must be cautious because consuming local food or water without taking precautions significantly increase their risk of infections. This highlights the importance of awareness and preventive measures for anyone traveling to high-risk regions. That's all from me. Thank you.